Bonjour. That's all the French you get from me. I work at the Buck Institute for Research on Aging in California. It's in Marin County, which is why I work with Fred Morin. Is that how you pronounce it? It's the same name. I think it's a coincidence, but I'm not completely sure. What I want to talk about today is therapeutic suppression of mitochondrial radical production. So before I can do that, I need to talk about radical production in mitochondria. So I'm going to tell you about the different sites in mitochondria that are able to make free radicals, to make superoxide and hydrogen peroxide, which in this talk is an honorary free radical. And then I'm going to tell you that even if you know what the sites are, they're very slippery. They change every time you look at them or every time you change the conditions. So the contributions of those sites change depending on the conditions. But under near physiological conditions, as best we can estimate, then 4 of those sites are dominant. So although there are 11 sites in mitochondria that can make free radicals, we're only going to concentrate on 4 of them. And in fact, I'm only going to concentrate on 2 of them because the therapeutic suppression part is that we have suppressors of electron leak at 2 of those sites, which are able to stop electrons from leaking onto oxygen to make free radicals. So we can use them as a scalpel to go in and understand the roles of those sites in different pathologies and physiologies. And I'll finish up by talking about various things that we might do therapeutically using these different molecules that we've discovered. So the general context of the talk then, the general idea is that life is bad for you. Life causes production of free radicals, of oxygen metabolites, causes oxidative stress, and that oxidative stress causes all of those conditions that we associate with aging. And the free radical theory of aging, which nobody really believes anymore, postulates that ROS production, reactive oxygen species production, by mitochondria is the cause of radical stress and all of these conditions. And although nobody believes that hard version of the aging program, the soft version is widely believed. And that is that radical production by mitochondria is an important contributor to all the nasty things that are going to happen to you over the next 100 years. So what reaction are we talking about? We're talking about putting electrons onto oxygen. And of course, that's what you are doing now. You're breathing in and out to take in the oxygen, to provide it to your mitochondria, so you can run these reactions. And the reaction you're trying to run is this one, where you take an oxygen molecule, you add 4 electrons and 4 protons, and make water. And in the process, a lot of energy is released, which you capture as ATP, and you use that to drive your incredibly acute brains, which are listening to what I'm saying. And that's happening 99.9% of the time. But occasionally, you can have leaks of those electrons. Either 2 electrons can leak onto oxygen to make hydrogen peroxide. In general, that's a bad thing. You don't want to be filling your brain with hydrogen peroxide. And then sometimes, even worse, you can put electrons on singly to make the superoxide anion radical, which is unmitigated bad news, I think, when you make that. It looks like what biology has done is to exploit these reactions which happen to do some signaling. So that's why it's only usually bad, because you get some signaling functions that come out of this system as well. But the radicals themselves, the superoxide radical or the derived hydrogen peroxide, are generally reactive toxic molecules that damage lipids and proteins and DNA. So we're interested in a number of questions. Why would you design a system that behaves like this? This just seems like a poor choice. So is it inevitable that electrons leak onto oxygen to make these radicals and cause damage? And the answer is no, it's not inevitable. It's the usual evolutionary quick fix, where evolution has decreased the radical production to the level that's acceptable and then gave up on the problem and went on to look at other problems. So we can do better than evolution. We can find small molecules that can improve the system and decrease these reactions. And then if you think about what causes the radical production in the first place, it's simple chemistry. We have some donor of electrons, let's call it X. So there's an X with a dot on it, an X dot minus, which can react with oxygen in a simple bimolecular collision reaction, presumably catalyzed by or associated with proteins, to form the oxidized version of X and the reduced version of oxygen, the superoxide. So if we want to know how fast you are making superoxide now, it just comes down to a simple equation of it depends on the oxygen tension at the sites in your cells where you're making superoxide, and it depends on this thing X. It depends on the redox state of the electron carriers in your electron transport chain as to how fast you'll be running this reaction. So one of the myths that I constantly run across is, particularly from physiologists, who think that the rate at which you're respiring will change the rate of this reaction, will change how fast you make radicals. That's a myth, and it's not true. It doesn't matter how fast you run electrons through the system. What matters is the steady state reduction of the intermediates. If they go more reduced at a given oxygen tension, you'll make more radical. And if they go more oxidized, you'll make less radical. So I'll keep coming back to that. The flows in this system are not what determines the rate of radical production. It's the potentials. It's the redox potential of the members of the electron transport chain. So here's the electron transport chain. I like to draw it this way because it shows the energetic relationships much more clearly than the standard models that you see of blobby proteins in a membrane, or even fine structured proteins with all of their alpha helices and other secondary structure in place. This is a diagram of the electron transport chain in energetic terms. So this is the energy scale up the side, more reducing at the top and more oxidizing at the bottom. These different planes are supposed to represent isopotential groups, where the redox potential of all the components is the same. So there are three main isopotential groups in mitochondria. One group around NADH and NAD, and this is where all of the standard Krebs cycle intermediates feed electrons into oxidative phosphorylation. Slightly more oxidized is the level of the quinone pool, ubiquinone, ubiquinol, where some substrates, such as succinate, which are not energetic enough to feed electrons up here, feed their electrons indirectly. And then cytochrome C is at a more oxidized potential, and oxygen itself is at the most oxidizing potential. So the way the system works is it's like a simple hydraulic analogy. You pour electrons or you pour water at the top of this fountain or this cascade, this waterfall. They flow around, they exchange between different intermediates, and then they find a hole, and they pour down the hole down to the next level. And as they drop down from one level to the next, you can harvest energy. You can put a little water wheel here and drive it as the water flows down from this higher level to the lower one. So you pump protons, and that's used to drive the ATP synthase to make ATP, and you extract energy from the system. So electrons would come in from Krebs cycle intermediates through these dehydrogenases into complex 1 of the electron transport chain, eventually drop down into the level of the Q pool to complex 3, and then the same action is repeated. You run down to the next level, get more energy out, run down to the next level, get more energy out, and eventually park the electrons safely on oxygen to make water. And one of the nice things about this diagram is it lets me explain to you about reverse electron transport. And reverse electron transport turns out to be a very important property of mitochondria in ways that we hadn't suspected. With reverse electron transport, what happens is you feed in a substrate at this level, for example, succinate or glycerol phosphate. They pass electrons into the Q pool, which then run down the waterfall, generating protomotive force or generating ATP. And what that gives you is a steady state where the quinones are very reduced and there's plenty of protomotive force. And that allows the protomotive force generating reaction in complex 1 to go backwards. And the electrons from here are pushed uphill to the more reducing state. using the proton force that other electrons have given up as they drop down the energy levels. So if you feed in electrons at the level of succinate, they can be pushed back by reverse electron flow into complex 1. And when they do that, they're a very potent source of superoxide and of free radicals that cause all the things I was talking about. So when I moved to the Buck Institute about 8 or 9 years ago, I decided I needed to, for my own calmness, I guess, I needed to be able to understand the different sites in mitochondria that electrons were being leaked onto oxygen to make radicals. And these red dots each represent a dead postdoc or PhD student. Each red dot is one of the sites where electrons are able to leak out of the system onto oxygen to make either superoxide or hydrogen peroxide, or sometimes both, and sometimes we're not quite sure which one it is. And we spent a lot of time on each one just trying to understand what are the properties of that site, what makes it run and makes it not run. And the equation I showed you before with x dot minus as the donor actually explains everything that's going on in here. X changes at each site, it's a different thing. Here it's the flavin in complex 1 at the site we call 1F for the flavin site of complex 1. Here X is a quinone in the other site in complex 1, the Q binding site. Here X is the flavin in complex 2, which is picking up the electrons from succinate. But in each case, that X, once it's reduced, has a finite probability of reacting with oxygen to make superoxide and to make radicals. So that's what each of the red dots indicates. So then the second question we moved on to is to ask, how do you measure how fast each of those sites is going? When you have a system with 11 sites, and when you think about electrons, electrons are badly behaved. They will tunnel under barriers. They will move around to different carriers as soon as you do anything to the system. So how do you find out which of those sites is making superoxide and how fast it's making it? And the problem is that because of that characteristic of electrons, if we just inhibit a site, for example, if we add rotenone to the electron transport chain, which acts there to prevent that site from running, the problem is that the electrons immediately redistribute. So if we add rotenone to block electron transport here, all of the electrons coming in from the Krebs cycle just back up, reduce everything else in the system, and these sites start to go faster because their X dot minus concentration increases. And conversely, all of these sites down here, if you're just running in a single substrate into the NAD pool, all the sites down here will go more slowly as the electrons in this part of the system drain out to oxygen, and these centers all go oxidized. So if you make a simple addition of an inhibitor of a site to find out how fast it's going, the biology defeats you because some sites in response go faster as the electrons go into them, and other sites go slower. So it becomes very difficult to interpret. And the literature is full of experiments where people add rotenone to cells, see some signal change, which they think is caused by complex 1 Q binding site making superoxide. The change is caused by preventing that reaction. But because of the confounding effects at all these other sites, it's actually really difficult to make that conclusion. And sometimes people find when they add inhibitors of electron transport chain, ROS production goes up. That's because these things go more reduced and produce more ROS. And other times it goes down. And that might be because, for example, this site might have been the original dominant site. And when you added rotenone, it no longer runs because it doesn't have any electrons. And the same criticisms apply to all the genetic approaches that have been used. People would make a mutation usually in complex 3 and find that the ROS production in their system had changed, but it's confounded by all these other changes. The electrons behave badly, go off in other places, and it's impossible to get an accurate description of what's going on. So how do you get around that problem? And in the end, we decided the way to get around it is to exploit that equation of X minus plus oxygen giving superoxide by just measuring X minus. If you measure the redox state of the thing that's producing the superoxide, then you can work out how fast that site's going just by calculating the rate constants in that equation. We can't measure the redox states of all of these things conveniently, but we can measure a couple of things. We can measure the NADH-NAD ratio very easily just by absorbance spectrometry or by fluorimetry. And we can measure the QH2Q ratio because it's in close equilibrium with the cytochrome B in complex 3, and we can measure that by absorbance spectroscopy. So we can measure the redox states of these different isopotential groups. That's the X minus in the equation. And then all we have to do is measure the rate constants, and then we'll know how fast any particular site is going under any condition. So this shows that measuring and calibration. So here's the NAD and NADH pool from this upper, more reduced isopotential group. We can use inhibitors to block off everything else and particular substrates, for example, malate, just to feed electrons into the system. And if we block off everything else with rotenone, we block off all of the rest of the chain. We don't feed in substrates that would do anything from these sites. We add inhibitors that stop them from working even if they do get some electrons. Then the only site that's producing superoxide is this flavin site in complex 1, which is in close equilibrium with the NADH-NAD pool, which we can measure. Then we can vary the electron flow, so we can vary the amount of malate that we have there, or the activity of malate dehydrogenase, and that will cause the NAD level to be more reduced or more oxidized. We can measure how fast the system is producing radicals, and that gives us a calibration curve in these blue dots here, which shows us how fast this flavin site in complex 1 runs as a function of the NADH-NAD ratio, as a function of this redox state, which is a function of the flavin redox state, the X minus in that site. You all look puzzled, but that's really powerful, because what that allows us to do is to have a calibration curve that tells us if we take some mitochondria in a brain, in a cell, in an organism, if we measure the NADH-NAD ratio, we can look up this calibration curve, and we could just read off and say, okay, that 1F site, the flavin site in complex 1, is making superoxide at a rate of 25 picomoles of superoxide per minute per milligram of mitochondrial protein. So we can directly use the calibration curve to say how fast is one of these red dots running. And then we can do the same thing for the complex 3 site. The quinone binding site in complex 3, we can measure the B566 redox state, which reports the redox state of the QH2Q ratio. We can use appropriate inhibitors to block off all of the other sites. We can vary the redox state of the B and the Q, plotted along the bottom here, measure how fast the system makes superoxide or hydrogen peroxide, and get a calibration curve. So now if we, in principle, but in practice we haven't managed to do this, if I could measure the cytochrome B redox state in your brain, I could tell you how fast your complex 3 is making superoxide, just by looking up this calibration curve. This, incidentally, assumes you're all rats and not people, because it was a rat system that we used to get the calibration. So there are obviously caveats to the things I'm saying, but the principle the principle applies. So we can measure how fast the sites go, and then we can run them under optimal conditions to find out how fast they can go, what's their maximum capacity. How fast can each of these 11 red dots make superoxide or hydrogen peroxide? If we go through each of the sites, the way we do it is to add various inhibitors and then correct for the electron redistribution using the tools I've just been through. We can say that actually the flavin site of complex 1, which is the one that the literature is most excited about, is the site with the least capacity in the whole of this first isopotential group. In fact, the oxyglutarate dehydrogenase has got something like 10 or 50 times higher capacity to make superoxide than complex 1 flavin has. The pyruvate dehydrogenase flavin is also very high, as some of the other branch chain keto acid dehydrogenases, the branch chain and other members of the group. So we can work out the maximum capacities of each of the red dots in this isopotential group. this Q site in complex 1, which is the one that runs during reverse electron transport, it turns out to have a much higher capacity than any of these sites. So that's one of the reasons I'm going to keep stressing it. When you push electrons back uphill into this site here, the quinone binding site of complex 1, it makes superoxide like there's no tomorrow. And of course, in some sense, there is no tomorrow if you make too much superoxide. Yeah, go. And then if we measure the capacities of the sites in the next isopotential group, the site with the biggest capacity, at least in rat skeletal muscle mitochondria, which is the ones we're using here, the site with the biggest capacity is the quinone binding site in complex 3, the outer quinone binding site, which has twice the capacity of any other site. And then surprisingly, complex 2 is a very good producer of superoxide. So complex 2 under optimal conditions, in particular you have to leave the flavin unoccupied by substrates, is very happy to donate its electrons onto oxygen to make superoxide. So the flavin site of complex 2 has high capacity. The glycerol phosphate dehydrogenase is also very good, but in rat skeletal muscle it's not a very abundant enzyme. If you look in other tissues like brown adipose tissue where it's more abundant, then the glycerol phosphate dehydrogenase becomes an important ROS producer. The ETF system in fatty acid oxidation and other minor enzymes are also able to make superoxide and radicals. And this is just to remind me to draw your attention to this part of the graph. This is the topology, and it's deliberately drawn in a very small scale. It's because the data is crap. And all I can tell you is whether most of it is produced to one side of the membrane or about half of it. And what you can see is that for nearly all of the sites, they operate almost entirely to the matrix. So the Krebs cycle dehydrogenases sit in the mitochondrial matrix. What that means is if they make superoxide by leaking electrons onto oxygen, it's in the mitochondrial matrix. It's not in the cytosol. The nucleus doesn't know about it. But if that superoxide gets turned into hydrogen peroxide, which it does in the matrix, then it can leak out of the mitochondria, and what the cell knows about is hydrogen peroxide produced by the mitochondria. The exception is the 3QO site. This is about 50% directed inwards and 50% directed outwards. So when you're on this site, you actually make superoxide into the cytosol. So it matters a great deal which site is making your superoxide or making your H2O2. If it's complex 3 that's making it, it can make superoxide directly into the cytosol. And you can well believe that that might be very different from if, say, the 1Q site is making it, when it's making it into the matrix and superoxide never appears outside. It's only hydrogen peroxide that appears. And I think that's very important in some of the consequences of running different sites, is the topology of the production. Okay, what I've talked about is capacities. How fast can we make that site go if we really push it? And if you read the literature, 95% of the literature only considers capacities. So people measure how fast you can drive ROS production from a particular site in mitochondria. But if we're honest about it, neither you nor I care about that. Who cares how fast it can go? What you care about is how fast is it going, because that's what's going to kill you, not how fast it might go. So what we're more interested in really is the native rates. So the capacity is equal to Vmax of an enzyme. It tells you the maximum rate it could go at. But the native rate is equivalent to V, the rate at which it's currently running. The capacities are very useful if you want to do enzymology or if you want to do screening, which I'll talk about later. But the native rates are very important for trying to understand what matters physiologically. So now I'm going to talk a little bit about the native rates. And the way we started doing that was the way that most of the field does it. We take isolated mitochondria, we present them with our favorite substrate, for example, in this bar, succinate, and we measure the amount of superoxide or hydrogen peroxide the mitochondria are producing. And when we do that, we can now assign the different sites. So we measure the redox state of the NAD pool, and look up the calibration curve, and what we find is that the flavin site in complex 1 hardly contributes to that rate. We can measure the B redox state in complex 3, look on the calibration curve. That site hardly contributes under these conditions. But if we block off the Q binding site in complex 1 by adding rotenone, correct for all the electron shifts that take place, which is, it turns out, not a large correction. And then we can assign almost all of the radical production from isolated skeletal muscle mitochondria oxidizing succinate to this quinone binding site. Most of the important electron flow is backwards flow from the Q level up into complex 1, causing leak of electrons onto oxygen to make superoxide. However, if we switch substrates to other favorite artificial substrates, the picture changes completely. So if we use glycerol phosphate as substrate, we still get some reverse electron transport, so we still get some of this 1Q site running. But now 2 other sites start to contribute. The glycerol phosphate dehydrogenase itself, which doesn't run until you give it electrons. But if you give it glycerol phosphate, it has electrons to donate. But also the complex 2 site, succinate dehydrogenase under these conditions. What's happening there is the electrons go into the Q pool from the glycerol phosphate dehydrogenase and then come back out on the succinate dehydrogenase and sit in the flavin site saying, here I am, take me, take me. There's no succinate sitting in the site because we didn't add any. So the only thing that can happen to that electron is eventually an oxygen will come in and pick up the electron and make superoxide. So under those conditions, the 2F site, the complex 2 flavin site, is a significant contributor. If we switch to fatty acids as a substrate, palmitoyl carnitine, the overall rate is less. And now it's the Q site in complex 1 doesn't contribute, slightly surprisingly. It's divided about equally between the 2F site, the 3QO site, and the 1F site. And if we use another conventional substrate, glutamate and malate, commonly used in mitochondrial experiments, we get another site running, the oxyglutarate dehydrogenase. The OF site runs and makes a significant amount of the superoxide. So there's 2 take-homes from this slide. One is how fast your mitochondria are making radicals, which are going to kill you eventually. Depends on what substrates they see, what metabolites they're being presented with. What you're eating may affect how much electron transport is diverted into superoxide production. So the absolute magnitudes are very different. And then which sites are contributing is also very dependent on how you put the electrons in. So what are you doing? Which of your sites are making superoxide as you sit and listen to me now? That's kind of a difficult question to get at. And we realize that what you have to do is you have to actually give mitochondria substrates that they would see in vivo. It turns out for the system we were looking at, which is rat skeletal muscle, there's a really good literature going back into the 1960s and 1970s where people spent a lot of time rapidly freezing muscle at different exercise states and then laboriously analyzing all the metabolites. So we can go back to that literature and work out exactly what is in the cytosol of a muscle cell in a rat when it's at rest, or when it's exercising mildly, or when it's intensely exercising. And we correct for the mitochondrial cytosol distributions, and we correct for the extracellular contamination. And when you do that, you can get a complex mix of substrates with different conditions of ATP and pH and magnesium, all of which are relevant to how fast those mitochondria will be able to take up electrons and generate superoxide and hydrogen peroxide. So this is the best mimic we have of what you're doing right now. Your muscle mitochondria probably see something like this. That's what they're doing at rest. Which of the sites is going to be running? So first of all, how fast are these mitochondria running? So isolated mitochondria in this mix, mimicking rest or mimicking exercise, run at a rate which is much less than the maximum capacity of most of the sites. So you can't say, well, the rate's up here, so it can only be these major sites. It could be any sites that are contributing. And then secondly, to our surprise, when you mimic exercise, the ROS production by the mitochondria goes down. And what the literature says is it goes up. But it's clear that it goes down. And from a bioenergetic standpoint, that's what it should do. When you go into exercise, the ATP demand goes up, electrons flow down the chain faster, everything goes more oxidized, so X minus, whichever X minus it is, drops, and therefore the rate of superoxide production and hydrogen peroxide production drops. So when you see increases in ROS during exercise, we think they must be coming from somewhere else. They're not coming from the mitochondria. If our mimicking of isolated mitochondria, using isolated mitochondria, if that mimicking is a valid measurement of what's really going on. So we can use the same techniques then to dissect what happens in this complex mix. And here's the answer at rest and exercise. We're now looking, remember, at these rather small rates, so the error bars are getting bigger and bigger. But essentially, at rest, only 4 sites dominate, or maybe 5. We think this one may just be error, which happened to be significant in our experiments, because when we try and drive the maximum rate of this site, it's less than we measured in these experiments. So we think 4 sites are running. The flavin site of complex 1, that's one of the traditional ones that you'll read about in the literature. The complex 3 quinone site, that's the other traditional site. But more importantly, or at least equally importantly, are the flavin of complex 2 and this reverse electron transport site in complex 1. So probably there are 4 sites in your muscle mitochondria running right now, which are generating superoxide. And then when you exercise, everything tends to go oxidized. The NAD pool tends to stay pretty constant, and therefore the contribution of the flavin site, which you remember was the one with the smallest maximum capacity, it just keeps plugging on, and eventually at rest, during exercise, it probably dominates what's happening. So the NAD pool is kept fairly reduced, and that means that during exercise, the flavin site in complex 1 becomes a significant contributor to your ROS production. So if you fell asleep, what I just told you was, of all those red dots, you can forget about 7 of them. There's only 4 of them that matter. 1F, 1Q, 3QO, and 2F. I only know that statement for isolated rat skeletal muscle mitochondria, but from here on in, I'm going to extrapolate it to all organisms and all tissues under all conditions. And clearly that's not going to be true. But it's a simplification at the moment because we don't have any better information than that. So there are 4 sites in the electron transport chain that are spilling electrons. That's what life is all about. And what they're doing is causing all this damage. So why don't you just go down to the pharmacy and buy yourself a bottle of antioxidant supplements, and then you will live forever? Well, that's what most people in California actually do. What the epidemiological studies tell you is that antioxidants don't work. Antioxidant supplements, in general, don't work. They don't extend lifespan. And in fact, in some studies, they actually shorten lifespan slightly. So antioxidants are a bit dangerous. Maybe that particular antioxidants at particular doses will do you good, but there's no good epidemiological evidence out there to show that. So what's wrong with antioxidants? I guess it's appropriate for Bordeaux to talk about red wine. This is an analogy that my wife came up with when I was trying to explain to her what I do. And I had a long 5-minute explanation of this argument, and she said, that's just like red wine on a carpet. And I reckon it is. So antioxidants are like trying to mop up the red wine that's spilt on your carpet. You can get rid of a lot of it, but you're going to be left with a red stain, whatever you do. And so antioxidants can never completely solve your problem. The therapeutic part of my talk is, well, what if you were to not spill the wine? What if you were to prevent the radical production in the first place? maybe that's going to be better. So maybe the problem with antioxidants is they come in too late, they're too unspecific, they work in the wrong place on the wrong species. All of those problems can be solved if you just don't make the radical in the first place. So now I'm going to talk about suppression of radical production by mitochondria. And it should be apparent from what I've been saying to you is that we have in place an excellent screen for compounds that would stop the red wine from spilling on the carpet. We've set up assays which allow us to measure the activity of each of those 11 sites. We now know, or think we know, that only 4 of them or 5 of them really matter. So we can use a simple fluorescence plate reader assay to ask, well, how fast are the sites going, and if we add stuff from a library of chemicals, can we stop it going? So we set up initially in-house a small screen of about 3,500 molecules and just asked, can we find a molecule which stops, for example, the 1Q ROS production, but doesn't do anything at any of the other sites? And rotenone is such a molecule. It will stop 1Q ROS production. So then we had a second requirement, that is, it's not rotenone. If you take rotenone, it may stop your radical production, but it would also kill you because it would stop your mitochondria from harvesting energy from electron transport. So we have to allow oxidative phosphorylation to continue. So we did a counter screen, which is the mitochondrial membrane potential, to screen out anything that interfered with oxidative phosphorylation. We thought this was a bit of a leap in the dark, that you would be able to find a small molecule that stopped complex 1 from spilling electrons onto oxygen, but didn't stop it from doing everything else it does. But it turned out you get lots of hits when you do these assays, when you do these screens. So we got hits for the 1Q site, and we got hits for the 3QO site. We actually did get hits for the other sites, but when we went back and validated them, the number of viable candidates decreased and eventually dropped to 0. So we ended up with no good suppressors of the other sites, but we ended up with very good suppressors of these 2 sites. So then we went back with a pharma partner, Novartis, and we tested their library of 635,000 compounds, running them through this screen, finding molecules that selectively prevent the 1Q site and the 3QO site from making superoxide. And it works really well. So we tried to think of a snappy name, and we ended up with this one, which it's clear is not at all snappy, for which I apologize. So these are suppressors of electron leak, SELs, and then the ones that inhibit the 3QO site are suppressors of 3Q electron leak, which we then convert into sequels. You'll see we came up with the sequel before we did the original, so it's a bit like Star Wars, this program. So these are 3 of the sequels we came up with, 3 different chemical backbones, all of which have that same property. They can prevent specifically this site and no other site from making superoxide, and they don't interfere with oxidative phosphorylation. In fact, they don't interfere with anything that we can discover in any system. When we find one that does interfere with something else, we scrap it. So we still have plenty of candidates left that have passed all our hurdles. So this just shows that. This is measuring the activity of ROS production from different sites against the concentration of each of the sequels. This is sequel 1. We can take out the 3QO site with about micromolar or half micromolar IC50, but it doesn't do anything to OXPHOS or anything to the other sites. So we have specific scalpel tools that can take out one of those sites and stop it making superoxide. That's wonderful. That means we can test all sorts of theories as to who's making the radicals under which conditions. And we can start thinking about therapy. If complex 3 making superoxide is what's killing you, sequels should stop that reaction and should be able to protect you. This just shows that when we measure their effect on oxidative phosphorylation, there isn't one. So the 4 bars are just the 4 different sequels, the control in 3 different sequels. This is under different respiratory states with different substrates, and each time all 4 bars are the same height. So we're not doing anything to oxidative phosphorylation with these compounds. And this is the cycles. These work at the other site, the 1Q site in complex 1, two different cycles shown here, cycle 1 and cycle 2, with different structures. Same thing, we can take out the site we're interested in without any effect on other sites or on oxidative phosphorylation. So we have a second scalpel that will take out the second of these sites that's able to make superoxide cycles. If you fell asleep, all I said was we were clever and we found some compounds that can stop this site from making radicals without stopping any of the regular stuff. And a second set of compounds, the sequels, which can do the same thing at this site. So can we use them for anything? Obviously, the answer is yes. We can use them for all sorts of things. And we've found, in general, that whenever we throw these molecules at systems, cell systems or whole animal systems, where people have postulated that mitochondrial ROS is doing something, then the compounds stop it, which is surprising to me in a number of ways. One is that really these people using these terrible techniques actually got the answer right, that mitochondrial ROS does matter. And these compounds really do get in there and stop things from happening. I'm going to give you one example from each of three different categories, which are kind of arbitrary categories. One is physiological. If we just take a cell and we're as nice to it as we can be, are the sites running and can we take them out? And the answer is yes. Signaling, so if we take systems that are known to be signaling, either what you might call normal signaling in response to hormones or other things, or pathological signaling, can we take them out? For example, HIF1 alpha signaling in hypoxia is postulated to go through complex 3 superoxide production. The answer is yes, we can take that out if we add the sequels, so we can demonstrate that that hypothesis is true. And then damage, overt damage. If the site is just running too much and causing damage, can we ameliorate that damage by using sequels and cycles? And I'm going to talk about ischemia reperfusion injury, where the cycles are very effective at protecting against ischemia reperfusion injury. So here's our topology. So remember, we have molecules that can take out the ROS at either site through QO, which is both sides of the membrane, superoxide to the outside of the mitochondria or to the matrix. So cytosolic effects due to superoxide alone will be sequel sensitive, but they won't be cycle sensitive because superoxide never comes out. And then the cycles and complex 1 Q site only makes its superoxide into the mitochondrial matrix, and the only thing that appears outside is H2O2. So by using the cycles and sequels, we can start to unpick for a particular process, is it driven by superoxide or hydrogen peroxide? And one of the things I've tried to do, I don't know how successfully as I talked, was to avoid saying ROS, reactive oxygen species ROS. I think it's a terrible thing to say because it doesn't define what you're talking about. And the different reactive oxygen species or radicals have different effects. And if you think in terms of ROS, you're going to get confused because one molecule will do one thing, another one will do a different thing. And there isn't, for example, a ROS suppressor. There's no such thing. There are only suppressors of hydrogen peroxide concentration or superoxide production rate. So this is the physiological example. This will take a moment just to explain. What we did was to take primary astrocytes from mice and to be as nice to them as we could. So we cultured them for a few days, 5 days or 3 days in this experiment, either under normoxia, atmospheric oxygen, which is supposedly a terrible thing to do to cells, or under very low oxygen, 3%, which is supposedly a bit more like the physiological. And then we just asked, is there any evidence of ongoing oxidative damage caused by superoxide production by the mitochondria under those conditions? The ways to measure superoxide and hydrogen peroxide are very poor, so we had to struggle with what our assay would be. But in the end, we decided to use succinate dehydrogenase itself as an assay. Succinate dehydrogenase has an iron sulfur center in it, which is very susceptible to superoxide, and it's nicely located in the matrix. So the activity of succinate dehydrogenase is a good surrogate measure of the superoxide concentration in the matrix. So normally it comes out at a normalized 100% value. But what we find is if we incubate the cells with cycles at different concentrations, we can increase the endogenous level of SDH activity. We don't change the amount of the protein. All we do is change the amount of that protein that has an oxidized iron sulfur center and gradually bring it up towards 100%. And when we do that, we get about a 50% increase in the succinate dehydrogenase activity. So what we think that means is that in these primary astrocytes, there's normally a production of superoxide, in this case from complex 1, but the same result is shown with complex 3, which is causing superoxide to be produced in the matrix, which is damaging the succinate dehydrogenase, and its level is depressed, and then presumably it's being turned over, the iron sulfur centers are being replaced. So you're in a steady state of damage and repair. And when we add the sequels or the cycles to suppress the superoxide production, we remove some of the damage, so the repair takes the level up, and we get an increased level. So my interpretation of that, and again it's a big extrapolation, is that in your brains as you sit here, complex 3 and complex 1 are making superoxide and they're damaging your succinate dehydrogenase. And we could stop that happening by feeding you sequels and cycles, and that presumably would be a good thing. Here's an example of the second category, the signaling pathways. Again, it's a relatively complicated model. We're looking at an endoplasmic reticulum stress of gut stem cells. So in insects, this is Drosophila, the gut, in response to various ER stresses, the gut stem cells hyperproliferate, and that turns out to be a bad thing. Eventually it causes the gut to collapse and the fly dies. So you can give flies tunicamycin, which is an ER stressor, and they'll die fairly quickly. And the mechanism is an overproliferation of the stem cells in the gut. So here's our control. Here's the tunicamycin, and the amount of proliferating cells of phosphohistone stain increases. But if we do that in the presence of our cycles, then we can prevent most of that proliferation. So in Drosophila gut, there's a signal coming out of the mitochondria from complex 3, which is saying proliferate. We can dampen that signal by using the cycles, or the cycles in this case. The sequels do the same thing. This is another model using a genetic mechanism instead of tunicamycin, just to show it's not drug specific. So our model here is that the ER stress signals in some way to the mitochondria. We're very interested in what that some way is, to cause complex 1 to make more superoxide into the matrix. Turns into hydrogen peroxide, comes out, does something, quite interested in what that is, and that causes the detrimental hyperproliferation in these cells. And here's the final example, the overt damage example. So this is an ischemia reperfusion injury model in a mouse heart in a Langendorf perfusion. So you can isolate the heart, hang it on a Langendorf apparatus, and it'll beat happily. If you then stop the perfusion, the beating stops. And then when you restart the perfusion, you get a reperfusion injury. And the heart will start to beat again, but less well than it did before the insult. So here it is in chart form. This is the rate pressure product, which is just the power output by the heart effectively. It starts at 100%. During this gray box, we stop the perfusion, so it drops to 0. And then at 45 minutes, start the reperfusion, and it comes back, but it's now damaged, so you get a much less response than you had to start with. If we add 1 micromolar cycle, that's the 1Q suppressor, just during this green bar, just during the point of reperfusion, then most of the signal comes back. In this experiment, not completely. In other experiments, actually, it comes all the way back. So we can very strongly protect the heart against the reperfusion injury caused by ischemia. So here's the model. Ischemia causes the mitochondria to have all these electrons. They don't know what to do with them, so they dump them on whatever's available, and what's available is fumarate, so they make succinate. And then when you reperfuse, that succinate suddenly runs, it pushes its electrons into the quinone pool. you get reverse electron transport into complex 1, superoxide production, and overt damage. And the cycles simply prevent that ROS production, but they allow you to burn off the succinate. So after a few minutes, when it's gone away, you can take the drug out and you still have perfectly functional hearts. And again, you can see the therapeutic possibilities with something which can protect against ischemic fusion injury. So we think in general that when life throws these insults at us, particularly of this form through mitochondrial radical production and hydrogen peroxide production, sequels and cycles are a way to stop that as potential therapies. And most of my time at the moment is trying to fill out those last 3 slides into different examples to really work out where we can be effective and what sort of pathologies we can treat using sequels and cycles. A bunch of sites make radicals. Electrons are slippery, so it's very hard to work out what's happening physiologically. But under physiological conditions, 4 sites seem to be dominant. And we can stop 2 of those sites. And when we do that, we learn a lot about the physiology, but we also gives us an insight into the pathology and gives us a potential therapeutic tool to stop that pathology. A lot of people have done the work. I just sit and drink coffee in my office. These are the people in Cambridge about 10 years ago who really started the project. These are the postdocs and technicians who worked in my lab when I first arrived at the Buck. And this is my current group working on these topics. And lots of collaborators from different institutes who've provided a lot of the experimental expertise that we need to do the experiments. And funding from a bunch of nobodies and OP2 to help drive the process forward. And thank you.
Thank you, Martin, for this excellent talk. I have a question about one of the statements on your final slide. You say that in physiological conditions, these 4 sites dominate. What about disease? Because I'm not an expert, but I'm assuming that in disease you can have up or down regulations of activity at some sites or even mutations. And you said if you touch one element, it will have repercussions to all other elements in the system. So are there diseases where these 4 sites may not be the dominant targets for therapy?
Yeah, absolutely. So in principle, of course, you're right. It may be that glycerol phosphate dehydrogenase is the site that's going wrong in some disease or other, in which case our compounds will do nothing. As I mentioned during the talk, actually what we find is the compounds do something. So we haven't yet discovered one where there's good evidence in the literature that mitochondrial ROS does something, yet our compounds don't stop it. So although there should be examples of what you said, we haven't found one yet. They must exist.
Hi, Martin. Back to aging. You think that the percentage of ROS release counts for how much in aging? Is it everything, or is it only a few pieces of it, compared to genetic, for example?
Yeah, what's the answer to aging? Difficult question. It's clear that the hard version of the mitochondrial free radical theory of aging is not true. The hard version is that mitochondrial ROS production is the sole cause of all aging. That statement is not true. There's enough evidence against it. But the inverse is also true, that it's clear that ROS production by mitochondria does contribute to a lot of pathologies associated with aging. The extent to which it contributes is hard to know. And the reason it's hard to know is up to now we haven't had the tools to really explore that question. I think one of the exciting things about our molecules is they provide the tools to answer that question, but we don't yet know the answer to it.
In other words, did you try that molecule, these 2 molecules, in your model of, what is this model, Born? Worms? Aging?
Yes, so one of the first things we did was to put them into worms to see what would happen. Nothing happened.
Okay.
It's hard to know what that means. Worms don't take things up, they metabolize them, etc., so we didn't find anything. We have actually found in flies, if we give flies a high, rich diet, which is suboptimal lifespan, then the sequels will protect and we can get significant protection against an ad lib diet in Drosophila. So, yeah, hard to know exactly how to interpret that, but it looks hopeful that it will do something to aging.
Thank you very much. Very clear. I have just a question. You mentioned at the beginning that this superoxide production or H2O2 production could contribute to signaling. So, side effects. Could you expect that this inhibiting this 1 and 3 position sites could have some alteration of physiological functions, or it is only
that's a worry that we've been concentrating on for the last 12 months. If we are successful in vivo in preventing complex 3 from making superoxide, will that actually have detrimental effects? And there's some evidence, for example, from C. elegans, that could be the case. If you really dampen down ROS levels in C. elegans, they don't live very long. So it's a bad thing to do. We haven't found anything that's bad. We know that we can prevent the HIF signaling system, for example, and there are examples where you would expect that to be a bad thing. For example, in the carotid body, you're using the HIF system to signal what the oxygen tension is in the blood, and dampening that signal would be a bad thing. But we see no evidence that actually that happens when we experimentally address it. But we are very aware that the way the biology looks like it's been developed is that you evolve in an anaerobic atmosphere, you bring oxygen in, so you have a problem to solve, and then biology has sort of tried to fix it as best it can with various protective enzymes and modifying the enzymes where the ROS are being made. But you just do it sufficiently to get through your reproductive life and no better than that. And then you use the residue as a signal which tells you whether your mitochondria are under stress or not. And that's the signaling that maybe we would take out that might be bad. So we're very aware of it as a problem. We haven't actually discovered it to be a problem, but we're still looking for that possibility.
I was just having a question about the specificity of your compounds in terms of the other sites in the cell. You've showed that you screen them through screening for sites in the mitochondria production chain. However, in a cell or in an organism, you obviously have many more different sites. How do you know that your compound is specific to the mitochondria at the site that you targeted, whereas in a cellular system you have more? Did you control for that?
In the end, you can't control for that, but yes, we've tried our best. So, for example, I didn't mention it, but a part of the counter-screening was cell growth. So we would run just a cell growth assay, and anything that interfered with cell growth was thrown out. So things that are overtly bad for the cell cycle, et cetera, got thrown out early. And we've now advanced the program, so we're putting them into animals, we're doing tox studies, and we're not finding nasty things happening. So there's no evidence for any off-target thing. But this is an impossible task to say that you haven't hit some reaction you haven't measured yet. So it always remains a possibility. But there's no overt problem.